Good morning, dear students. I am Ram Singh Meena, and today we will discuss about electrochemistry important topic, which is Daniell cell or electrochemical cell. Let's we start. First of all we should talk about cell. What is cell? Then cell is a device which can convert electrical energy to chemical energy and chemical energy into electrical energy. Cell is a device which can convert chemical energy into electrical energy and vice versa, and electrical energy into chemical energy. Then we can say that cell are the device which can convert chemical energy to into electrical energy and electrical energy into chemical energy. And today our today topic is electrochemical cells, which is a part of cells. If we can divide further classify cells, then there may be two types. First type is electrochemical cell, and second one is electrolytic cell. Then what is the difference between both? Electrochemical cell can convert chemical energy into electrical energy, and electrolytic cells can convert electrical energy into chemical energy. Dear students, we can talk about this electrochemical cells. Electrochemical cells are those cells which can convert chemical energy to electrical energy. And electrolytic cells, those cells which can convert electrical energy to chemical energy. And now we can understand about cells and what are the categories of cells, which are electrical energy and chemical electrochemical cells and electrolytic cells. And today our topic is electrochemical cells. Then we start electrochemical cells. And look at the board. Today we will talk about electrochemical cell. Electrochemical cell. These cells are also known as galvanic cell. These cells are also known as galvanic cell, and also known as voltaic cell. And other one name is Daniell cell. It can maybe different names, which are electrochemical cell, galvanic cell, voltaic cell or Daniell cell. Now we can understand about this. We have two containers in which first container contain Zn rod, Zn metal rod, which acts as anode, and this rod is placed in aqueous solution of this metal, which is ZnSO4, salt of this metal. And like this, the other container contain copper rod, which acts as cathode, and this placed in CuSO4 aqueous solution. Dear students, there is U-shaped glass tube which placed revert. These are cotton cocks, and inert electrolyte is filled in this U-shaped tube. Both electrodes can connected by conductive wire, and this is voltmeter, and there is switch. If we talk to about salt, this U-shaped tube, this act as salt bridge. And now we can talk about some important things about this salt bridge. First of all, we should remember that this contain inert electrolyte. This contain inert electrolyte plus agar solution. Agar solution is nothing but paste of polysaccharides. Agar is paste of polysaccharides. Paste of polysaccharide. And inert electrolytes like KCl, KNO3, NH4NO3, these. If we take KCl as an inert electrolyte, then we cannot use these metals electrodes Hg, Ag, Tl, Pb, because KCl can form precipitate with these metals, and precipitate will be settled down and reaction will be not possible. Now dear students, we can talk about cell reaction. Then first of all, Zn as act as anode, that's why at anode, is it visible? Yes. At anode, Zn metal gives 2 electrons and convert into Zn plus 2 ions. These electrons from here, these electrons move towards cathode, and we obtain potential difference in this voltmeter. And if we talk to about flow of current, then we know that current is flow of current in opposite direction of electrons. That's why direction of current will be in this direction from copper cathode to anode. Then I have already mentioned that two electrons flow from anode to cathode. So these two electrons reach out at copper, means cathode. So there will be occur this reaction at cathode. Cu plus 2 aqueous plus 2 electrons gives Cu solid. And we can write net cell reaction. The net cell reaction, the net cell reaction will be Zn plus Cu plus 2 electrons are cancelled out, product side Zn plus 2 plus Cu solid. This will be Daniell-Sell's cell reaction. If we talked about cell representation of Daniell cell, before that we should talk about functions of salt bridge. Then what are the functions of salt bridge? Sometimes students experience the learning of anode and cathode, which one be cathode and which one be anode, and cathode will be positive or negative or anode will be positive or negative. Then we can use this trick. You can also form new trick of you, but I can tell you simple trick, which is lone. L stands for left, O for oxidation, A stand for anode, and N for negative, which means Zn, which is present at left side, which form Zn plus 2 ions by releasing of 2 electrons. So there is 0 oxidation, there is plus 2 oxidations, that's why oxidation. Okay, left oxidation, A for anode, means Zn act as anode because it present at above side in electrochemical series, and N for negative. And if we remember about this, then second one is cathode, which is copper, which will be right side of the cell and will be positive, and there will be reduction because copper plus 2 ions receive 2 electrons and gives copper, which means plus 2 0 oxidation, then there will be reduction. Then now we talk about functions of salt bridge. Function of salt bridge. First of all, salt bridge complete internal circuit. First, salt bridge complete internal circuit. Second one is it maintain electrical neutrality of both side solutions. like this. This is inert electrolyte, for example KCl, which gives K plus Cl minus. When we select inert electrolyte, then we remember that ionic mobility of cation and anion of inert electrolyte should be approximate equal. Ionic mobility should be approx equal. This is the first criteria of selection of inert electrolyte. We know that when reaction is proceed, there is Zn, this is ZnSO4, there is copper CSO4. We know that when reaction proceed, then in this side concentration of Zn plus 2 ions is increases and sulfate ions is decreases, and in this side concentration of copper plus 2 ions decreases and sulfate ions increases. It means in this side positive charge will increase and in this side negative charge will increase, which can create potential difference. That's why we use inert electrolyte. If there is positive charge increase, then this Cl ions, chloride ions move there and neutralize them, and potassium is move in this direction and neutralized negative. Like this they maintain neutrality of both side solutions. And last one function is, which is third, to stop generating of liquid potential junction. What is this? Let us explain it. For example, there is no electrolyte. For example, there is no electrolyte in this salt bridge. If there is no electrolyte, then we know that positive charge increase here, this move from here, and we know that negative charge increase here and this move towards. At this point, two opposite charge carrying points are generated. Due to this, in this point there will be generating of potential, which is known as liquid potential junction, and it can stop by the filling of inert electrolyte in this tube. Do you understand, students? Then we can say that functions of salt bridge are, first one is salt bridge complete internal circuit. This is internal circuit. Second one is it maintain electrical neutrality of both side solutions, and third one is to stop generating of liquid potential junction. I hope you can understand easily this. And now we talk about cell representation of Daniell cell. cell representation of Daniell cell. Then look at the board, dear students. First of all, we should write down anode, then product of anode with its active concentration. For example, in Daniell cell we take 1 molar solution. It may be variate. These two lines represent salt bridge. This represent anode. This salt bridge. And this is cathode. Sometimes we faces some problems like in given question we obtain cell representation and we need to prepare cell reaction. Then I am telling of you that how can we form cell reaction by cell representation. Then look at the board. This is left, this is right from this center. This is left, this is right from this center. You can stand like this. Left connect with left, this left is added with this left, and this right is added with this right. I want to say that, dear students, we can combine this left with this left and this right with this right. Both left can connected and both right can connected, and we obtain final cell reaction. Let's see. Left, this left, this left, this left. These are reactants and products, right and this right. And we know that this is net cell reaction of Daniell cell. Like this we can obtain cell reaction from cell representation. Dear students, today we talk only about these things. Next lecture will be tomorrow. Okay, thank you.